Yeah, that we can move to that next لو مفيش حد عنده سؤال. يعني دي ممكن نعمل ترانزيشن للفاكسين لو في حد عنده
شو
سؤال.
لو حد عنده سؤال يعني لازم يتسأل دلوقتي، ممكن تبعته على الشات.
تمام. في سؤال ولا جو ahead؟
يعني let's go ahead، ولو حد سأل
خلاص.
لو في سؤال ثاني. ده حد بس بيعتذر عن الاكزit. مفيش مشكلة، هيبقى في ريكوردنج ان شاء الله لو حد مش قادر يكمل. اتفضل يا دكتور علي.
دلوقتي لو احنا فكرنا ان احنا نعمل ستادينج للفاكسينيشن، واحنا بنعمل كده فعلا من قبل حتى كوفيد. افتكر السلايد اللي انا كنت بقول لكم فيها في الاول على موضوع الشورت ليف بلازما بلاست والجيرمال سنتر اللي بتحصل في الليمف نود التريننج، وبعد كده البون مارو. بس انت في هيومن انت عايز تبص في 3 بارتس. عايز تشوف الاول حاجه ايه هي الفيرست لاين الفيرست ريسبوندرز، ودول هتقدر تجيبهم في البلاد فيري ايزلي، وبيحصلوا دايما بيطلعوا في الاول usually within the first week or two. So you can capture that in blood. But then if you want to really have a look inside the kitchen, inside the real, the operation room in a way. This is where the whole الحرب كلها بتدار من الموضوع، من الأوضة دي، من الليمف نود.
غرفة العمليات.
غرفة العمليات، هي دي الكلمة، غرفة العمليات بتاعتها. هي دي الجيرمال سنتر. بس عشان تعرف تبص لازم نوصل ان احنا ناخد سامبل من الليمف نود، وفي الحالة دي الليمف نودز اللي
بس يعني حضرتك برضو انت قلتها طبعا، بس يعني اكيد في سبب ان ما حدش عمل الشغل ده لمدة 120 سنة زي ما انت قلت، ان هو it's not a piece of cake. يعني مش زي عينة دم كده بنسحبها، اي حد بيعرف يسحب دم.
الليمف نودز اللي موجودة مثلا، اللي لو احنا كنا بناخد الفاكسين في الابر ارم، الليمف نود دي كلها في الاكسيلري، والاكسيلري ريجن ده فيه ليمف نودز كتير زي ما انت شايف، زي ما شايفين في الدياجرام. ازاي تعرف تسامبل النود دي؟ اول حاجه دي محتاجه a very experienced.
وبرضه مش كل الناس هتوافق طبعا. فولنتير اكيد العدد بيبقى اقل من اللي يقدر يدي دم يعني.
بالظبط ان هي it's definitely not as simple as just going for a blood draw. You have to have IRB approval, and انت people get lidocaine first, like a local anesthesia, and then you ask them, you have an experienced radiologist, someone who can actually use ultrasound to sample those lymph nodes.
يعني قبل ما نترانزيشن الفاكسين، في سؤال انترستنج جه من الدكتوره ريهام، واعتقد هو سؤال جنرال برضه. هي بتسال لو البلازما ترانسفير، يعني البلازما ترانسفير، طبعا البلازما بتحتوي على الانتي بوديز دي، بس احنا كلنا عارفين نتائج نقل البلازما كان فيها مشاكل بسبب يعني ان البلازما بيبقى فيها فاكتورز ثانيه، ففي ناس بتنقل بس الاميونوجلوبولينز IVIG. فالى اي مدى البلازما ترانسفير از افكتيف يعني as a protection or treatment?
That's a really important question. Obviously, the plasma transfer or the serum therapy, that يعني just a side information. لو بصيت على the very first Nobel Prize for physiology medicine, it was 1901. الواحد العالم الألماني von Bern, and basically that's what he discovered, serum therapy. في ساعتها كان على الدفتيريا, treatment of diphtheria and tetanus, maybe diphtheria only. It's a very old treatment, and it's like the oldest trick in the book, which is really take antibodies from survivors and give them, in that case not antibodies, take the whole blood and send it. It's a very crude treatment, and really the answer for why it's not really the best one is this. You every one of us will be different, so when you take serum from all those individuals, all of those are survivors. So you can really, it's a very heterogeneous population of antibodies that you're transferring. So they are not really they're not very efficient. It's a great in the early stages of pandemic when you don't have all the antibodies. You don't have the refined monoclonal antibodies that you can use for therapy. Like you heard about Regeneron, we heard about other companies that made these refined antibodies that we know specifically are very potent, and you can make them as drugs, purified drugs, and give them as infusions, and that's way more efficient. So I think the serum therapy is really outdated, and I think it's very good in the very early stage of the pandemic. After that, quickly you transfer to monoclonal therapy. If you really need to give passive immunity, you go for monoclonals. And part of our lab is actually working on this. I have a part at the end if we have time, but this is something that we are actively doing. We actually, I can explain how we actually making antibodies from responding B cells and transforming those antibodies into therapy. And now we are working with pharmaceutical companies that I cannot reveal names, but they are very, very well-known big pharmaceutical companies who can actually do the bigger trials. Take these antibodies, make them as clinical material drugs, and then now they can be injected, and then they can be tested in humans. Academic labs cannot really make that transition. So usually the way it works in the US and basically everywhere is that you have academic lab can make the initial discovery, and then you have adoption by a big pharmaceutical company that come and just take the product and then basically take the whole thing. It costs millions of dollars to actually be able to do all the safety trials, FDA approvals, all of that. That's what big machines are good for. They can actually have teams that can lead these efforts. The labs usually, the rule stops at that point, and then we just become collaborators. So that's obviously a very important question about the same therapy. So looking in the غرفة العمليات في الجيرمال center, you can actually look by drain lymph node. You need a trained radiologist, you need ultrasound, and you actually have to have a team, a clinical team who can help you really make that possible. And this is one of the things that we were very lucky here. لما جيت هنا في الجامعة, the very first thing I did is actually establish that clinical team, because I really wanted to look in germinal center. And everyone is looking blood. Why don't we make the next step? So I have that team set up even before the pandemic, and this is very, very important. Obviously, it's very hard in the middle of a pandemic to set up something unless you have it already set. That's what helped us a lot, in that we had that already set. And then the last point, if you have access, and we do have access, you work with oncologists. And oncologists are very good at sampling bone marrows, because they do a lot of and I'm sure في مستشفى 57 الموضوع ده very common, and you sample the bone marrow by bone marrow aspirate for transplantation, for stem cell. I don't know if they do the hematopoietic stem cell for some sort of cancers. The treatment is you wipe the whole immune system. Some lymphomas, you have to wipe out the whole immune system and give a donor bone marrow. So all of this involves taking and injecting taking cells from the bone marrow. So I have a different clinical team, oncologists that help me get the bone marrow. So I have people in infectious disease who help me get the blood, people in radiologists that help me get the lymph node, and then people oncologists that can actually help me get the bone marrow. So the most critical thing in all of this is how to actually manage the team and keep everyone motivated, happy, and engaged. But also you have to make sure that they are all have invested in this. It's very important to actually make sure that they are really excited about why this is important. Remember, radiologist doesn't know anything about B cells. Infectious disease person doesn't know details about germinal center. An oncologist doesn't care about any of those things. Every one of them has already a set of patients that they are treating. So the most important the most important point is to make sure that to explain to them, to tell them, this is the big picture. This is why this is important. This is why the results from this trial will be transformational, not only for immunology, but also for anyone interested in vaccines. Anyone. So then they become really excited, and they will take from their heavy clinical duty, they will take some time and help you in that. And that for me was really priceless. I mean, you cannot put price on this. You cannot really give them money to do things. You have to give them the money to do things, but not only money that can make things work. And you have to give them that level of excitement and that level of engagement and investment. And this is important. Give them the vision that this is how we are going to understand how vaccination works in humans. Tell them that this is something that we haven't looked at it in humans before. No one on the planet is doing this. So this is what we are doing. And you can get wonders after that. You just keep the engagement and then move on the next level. Tell them why this is important. And of course, share the credit with them. And this is also very important. Yeah, you can, and this is something I learned from my mentors, Rafi, who is Dr. Rami mentioned. It's very important to actually appreciate and share the credit and acknowledge all the contributions of everyone. Science is not a thing, it's not a one-man show. And I'm gonna I'm just a representative from a big team, and that team is actually doing everyone in this team is contributing a very critical piece that if you remove that piece, the whole thing doesn't really fit. So you have to make sure that at the end of the day, you can actually go and say, thank you for everyone. And obviously in our academia, that means co-authorship and publications, shares in grants and funding. And in general, you actually help them because they also have careers that they need to further. And that's a very critical thing that I learned from my mentor, my postdoc mentor. It has nothing to do with science, but has to do with really how to not only start a big operation, but how to make sure that once it's started, how to keep everyone engaged and really feels that this is not a job, this is an investment. So قبل نتكلم عن RNA vaccines, أنا عايز بس I just give you an idea about when we started making vaccines. If the very first formal vaccines, obviously the smallpox. If you remember the very old story, the cow maids that were milking the cows, and they have the cowpox. The cowpox is a very similar. It's the same family as the smallpox, the Goodary. It's just attenuated in humans. If you take the same virus, same family, but it's actually the host of it are cows instead of humans, it's a very attenuated, and that's really how most vaccines are made. اللايف بوليو اللي هو شلل الأطفال اللي هو النقط اللي نديها الأطفال عبارة عن فيروس اتحط في درجة حرارة تخليته attenuated، يعني عملنا جروس لنفس الفيروس في درجة حرارة أقل من درجة الحرارة بتاعت جسم الإنسان. فعما تقعد تعمل له propagation في درجة حرارة أقل، فالفيروس acquire mutations بتخليه adapt لدرجة حرارة أقل. لو تاخد البرودكت ده تحطه في الإنسان بيبقى much weaker virus، لأن دي it's a different temperature, so it takes the virus time, and then this is how we actually we present to our immune system a much weaker version of the virus or the bacteria that our immune system can handle very well and develop the immune memory that we discussed. But now these approaches are outdated now, and we have been doing this for decades, and what's coming more and more is we have to be more refined. Why do we need to make an immune response to a whole bacteria or a whole virus? There's a lot of proteins, internal proteins, like here inside the virus, that are really important for the virus, but our immune system to these proteins or our antibodies against these proteins are not as important because they don't interfere with the very critical step of the entry. Remember when I talked about the very attachment step? So that's where more trend in the last 30 years is to really more minimalistic approach, which is go for that important target and make that as a protein. And that's how what we call subunit vaccines. So basically just take recombinant protein, and for example, in influenza, take the only HA and express that and give that to people. Why do you need an antibodies to the rest of the virus if you can actually, these are the most protective antibodies? And because protein purification and mass production is becoming a problem, as we are seeing now, why don't we make the protein outside and purify it if we just give the genetic material, which are the DNA, or now more recently the RNA, to our own cells and ask our own cells to make that protein? And then our immune system will quickly recognize that this is a foreign entity, even if it's made inside by our own cells. But our immune cells are not trained in their developmental stage to recognize that protein as self. And this is the very basic way of how we recognize non-self protein. and now they will deal with it as a foreign protein, and then immune response will follow. So how RNA vaccines work, and this is really the topic that we have been very focused on. I think RNA vaccines are really going to be an important component for many future vaccines. I can tell you that probably in 2 or 3 years الفلو شوت اللي احنا بناخدها دلوقتي غالبا هتبقى RNA. The trials already started. This is really not public information, but trials already started. There will be RNA vaccines based on influenza. There are RSV vaccines. There are even malaria vaccines.
تقريبا هيبقى كوكتيل يعني multiple vaccines.
Yes, there is a huge advantage, and then you can prepare mass amounts very quickly, and you can change that very quickly. So there's a lot of advantages that protein production just takes a lot of time. And as you can see, right now we don't have a single vaccine that's approved that I know of that's actually based on protein. You have the vaccines that are coming from the very early vaccines that came from China. They are based on whole inactivated virus. So we took the whole virus, inactivated the whole virus. And now that's why these vaccines are not very strong, because you are actually not focusing the immune response on the spike. You are giving the whole vaccine. The other vaccines that we know are the vectors, the adenovirus vectors. This is basically the Russian vaccine, the AstraZeneca, of course Johnson & Johnson. And again, you take the spike, put it into an adenovirus that can express that spike, and then that virus will you can actually generate a ton of that virus and then use it for vaccine. And obviously the RNA. I don't know if there is any other platform, but there is a company that's called Novavax that have been trying for 2 years now to make a recombinant spike protein. And it's already tested in phase 3, but it went through a lot of troubles.
لسه داخل مش كده؟
Yeah.
لسه داخل for approval because
Exactly.
of the adjuvant. I think the adjuvant is a problem, صح؟
That's a great point. The advantage, you don't want to give a very weak protein. If you just give a protein by itself as a vaccine, it's very weak. لأن basically أنت محتاج حاجة مع أي infection أو أي vaccine عشان الفاكسين يبقى successful. ليه whole virus is more successful than just give a single protein. لأن whole virus فكر فيها كده كأنك بتدي واحد أكل من غير أي spices، من غير أي بهارات، مسلوق. تحس إن هو there is nothing really in it. I can't believe it. I just think about adjuvant as the spices that you can actually just generate a lot of heat, a lot of excitement in our immune system, that the immune cells will be coming to the site where this new thing is. There's a lot of inflammation, and inflammation is actually, in a way, if it's a controlled amount, is actually good because you recruit cells. You have these cytokines that can actually almost like guiding for immune cells. بتلم الناس، والناس في الحالة دي immune cells بتحطهم في نفس place where you have your antigen, and then you have the processing starting and you have your immune system. And the more people you engage, the more successful, robust the immune system. And that's why recombinant vaccines you need an adjuvant. An adjuvant creates its own issues. And that's another advantage for RNA vaccines is that they are self-adjuvanted. You really don't need to have an adjuvant. They are enclosed in lipid nanoparticles, and طبعا كلية الصيدلة عارفين lipid nanoparticles much better than I will ever do. But you put them in lipid nanoparticles, and these nanoparticles can actually indeed be by themselves very immunogenic. That's how that will work, and it has been working. Some people find it more inflammatory than it needs, but that can be tuned down, and you can adjust the dose. You can modify the RNA vaccine itself, and we know that there's some significant modification to basically almost dial down the inflammation. And that's amazing that you can actually play with that in a way. So RNA vaccines are working in a very simple way. We give the vaccine, which is basically the main message for our ribosomes to translate the protein and we will just include a transmembrane domain, so that protein expressed on the surface. So you inject the vaccine, our immune cell could be a muscle cell, could be an immune cell, can take the protein, take the RNA inside, express the protein on the surface, and now we have a protein that our immune cells recognize as foreign, and after that, this is the RNA taken inside the cells. The spike protein is made outside. Our immune system recognized this and made the antibodies, and this is how immunity works. So now we wanted really to track the whole process. I call it the life cycle of a B cell, from the very early induction as a plasmablast, short-lived cells that make the first line of antibodies, as it goes to the operation room training to get the germinal center and lymph node, and then as it تطلع المعاش, mature, become really the long-lived plasma cells that live in our bone marrow. To see the full picture, this is what we brought to the field. We came with the team and said, let's study how these new vaccine platforms work in humans. And that's what I will spend the next few slides really explaining. This is the study we did here in WashU, and Washington University, where we do all our clinic study. So we enlisted the very first individuals that were approved to get the vaccine. And in the US, those individuals were healthcare workers, and told them, we need your help because you are fortunate enough to be offered the vaccine. So I basically told them that you need to help us understand how this vaccine works. And they were very nice and very generous, I would have to say. But we have a list of all clinical healthcare workers who actually told us they will give us blood, give us what we call fine needle aspirate. This is from the lymph node, and give us bone marrow at multiple time points after vaccination. And that's how we can track the journey of that B cell that got first engaged and then started the differentiation to an antibody producing cell, and finally as a bone marrow plasma cell. It's a journey. It's actually, you can think of it as a lifetime of experience the cell is going through. And it starts with literally a naive cell and ends up with a very, very experienced cell. And I always think of B cells as the trip or the journey that every one of us who actually go through their lifetime. They start very naive. They start to learn from people around them. And some of us travel, some of us don't travel, but we go through different experiences. We interact with different people, and we accumulate those experiences over time. And we reach to a level where we are really mature, where we really realize that what all life is about, in a way. But this is how our immune system, our B cell, go through that journey. And we were able to track that in multiple different compartments in our system. And that's what we will look at. So I just want to really, the question is, if you have been infected before and got the vaccination, how different you are if you haven't been infected before? And it's a very simple question, but remember, when you, is it a question?
لا خالص، هو كان يعني في سؤال اتسأل في النص انت هتجاوب عليه دلوقتي غالبا، اللي هو الفرق بين يعني التوتال antibodies والنيوتراليزنج antibodies. I'm sure you will address this later.
Oh, yes. So total antibodies, that's actually important. This is total antibodies. Basically, you look by how much antibody you are making against the spike, and then how much they're increasing. Remember, Pfizer is actually 2 doses that are giving 3 weeks apart. So after the first dose, you make some antibodies, and then after the second dose, you make more, and then you start the slow decay. For people who have been infected before, you make that antibody, and then after the first dose, you already have some from the infection, but then you have nice increase, and then the second dose doesn't add much. So the question about the spike, very important. So it's actually in the third part, but spike is not all the same. Remember, if you actually go to that part about the infection, here, it's very crude here, but literally there is a part in the spike which is the part that really get really the interaction with the ACE2, and that part we call it receptor binding domain. So it's a large protein that has multiple domains in it. So only that part, if you can actually have an antibody target to that part, So you can literally very physically block that literal interaction. That's the best antibodies. And usually those are the antibodies that are Regeneron or other companies are using for therapeutics because they are very potent. You don't need much of them, many of them. You don't need a lot of antibodies to block the infection. You can bind to the spike protein in different parts. So there's the internal domain, there are multiple other parts. Some of them can be neutralizing because the antibody has a footprint that can actually block the interaction, interfere with the virus. Not as potent. So there are antibodies that are in the last part of the spike, and usually those are not neutralizing. But our immune system doesn't really have a way of saying, oh, I should just make antibodies that against neutralizing or non-neutralizing epitopes. It makes the whole thing is foreign. We make antibodies to any part that we can target. You are basically shooting an elephant. You know that the brain is one shot in the brain will kill the elephant. But if you are in a really rush and you're just shooting, you shoot everything. Some parts will take through, really doesn't affect the elephant in a way. Some shots will go to the back leg and it just doesn't really hurt. It hurts, but it doesn't really block the rushing elephant toward you. So I don't know if that works. But so really it's how we can look at vaccine responses. These are antibodies. And of course, neutralizing will be a fraction of this. And some people...
الأحمر دول اللي ما كانوش infected قبل كده صح؟ والأسود اللي كان infected.
بالضبط. And you can see that the red ones are starting from baseline and the black one are higher than baseline because they had this previous infection. So they had some antibodies. They went through the whole cycle. So they have long-lived plasma cells. Actually, they are part of the study that we looked at their bone marrow plasma cells. And now they are getting first dose and you can see their memory cells gets really engaged. And then you have a lot more antibodies now because of that. But their second dose is almost really didn't make much because they have already enrolled.
معلش هقطعك تاني بس هي برضو انا بجمع الاسئله اللي بتيجي وبسالها في ميعادها يعني في سؤال اتسال واعتقد كل العالم بيساله برضو how much is enough يعني مين اللي بيقول يعني السؤال كل الناس بتساله طب انا قست الانتي بوديز اللي بترياكت مع سبايك لو 100 مثلا يبقى كويس ولا 1000 وهل كل اساي بيطلع نتائج متشابهه اصلا ولا التايتريشن دي مختلفه
سؤال مهم جدا. So اغلب diagnostic tests are based on binding. So they are really testing what we are testing here is if you have the spike, how much antibodies are binding to the spike. The 99% of those assays doesn't have anything about neutralization. You could have 90% of your antibodies for some reason or another binding to parts that are not neutralizing. So whatever value you have from that assay are meaningless in terms of protection. The ones that are making at least the most very clear correlate of protection is neutralizing antibodies. It's much clearer. Obviously, you will, if you have high number of antibodies, by probability, you will have a probability of higher fraction of neutralizing antibodies. That fraction is variable between individuals. But let's say on average, and this is like an assumption, let's say like you have 20% of every one of us has 20% of antibodies are neutralizing. Again, that's gross, very approximation. Then if you have 1,000 antibodies, 200 of them are neutralizing. If you have 100, 20 of them are neutralizing. So having higher antibodies will always be better because the fraction will be higher.
بس I think there is no magic number صح؟ يعني ما حدش لغاية دلوقتي وصل
At this stage, they are very close. There are some papers will be coming out in the next few weeks that are starting to really narrow down on how much neutralizing antibodies. المشكلة هنا وانت قلتها في السؤال المشكلة في الأساي. لو people for Australia, people in Europe, and people in US, and people in Africa, everyone is using different assays. So how you can actually normalize that? So there are a group of scientists, a task force basically, who sat down and basically took all the data and took different sera. So they took sera group and then distributed the same set of sera to different research institutes across the globe, and then asked them measure that X value, and then they actually now and use the same assay and giving the same all reagents, and they are now coming up with a normalized value that's going to be like some sort of called international unit, and it should be out in the next month or two. So it is coming.
من عندك ولا من لاب تاني؟
No, this is a much bigger offer. This is a WHO-led effort, and the main leading lab is actually in Australia. But yeah, this is a much bigger. This is an epi study. Our lab is not really doing the epi kind of studies. This is like the study where you have thousands of people having strong serum blood, and this is a much higher throughput that we can do. It's very important, obviously, because we can get this magic number that you just asked about. So everyone can look at blood and look at the antibody responses in blood. But then how you can actually now look at for غرفة العمليات, in the real operation where things are happening. And to do that, this is where we have the radiologist help us to really get the lymph node. So how you can get the lymph node. الأول عايز أعرف البروسيدور ده بيتعمل في مصر، أنا متأكد مليون في المية بيتعمل في أي مستشفى كبيرة، لأن إن أنت لو عندنا لو في حد عنده breast cancer, the very first step عشان to assess breast cancer, to assess the metastasis, the potential إن إحنا يكون الخلايا الكانسر نفسها اتحركت للليمف نودز، بنعمل أنا متخيل إن هو treating physician أو الدكتور هيوردر إن هو يتعمل كات سكان، هتعمل طبعا أشعة، بس بتعمل إن أنت ممكن تاخد عينات من الليمف نودز اللي هي draining the breast area for example، علشان نشوف إذا كان في خلايا transformed هناك، ولو هي خلايا، لو حد هيعمل العملية اللي هي removal, they usually take they also remove the lymph nodes علشان ما يبقاش في الخلايا الكانسر ما تروحش للبونز وتروحش للother organs حتى بعد ما تشيل the main primary site of cancer. So that process happens. لو احنا شاكين ان حد عنده lymphoma, you can look in blood, but actually the best site to look at are actually lymph nodes. So that's how radiologists actually know this process of sampling lymph node before even anything. It's a diagnostic procedure. The only thing that the innovative part that we did is we told them we want to do that diagnostic procedure when people had got the vaccine. So they are not patients, they are healthy volunteers who actually just got the vaccine. So can we actually sample their lymph nodes just to be able to look at the germinal center, to look at the training camp there, and then look at the cells that are being prepared for our future, for our memory. So the critical part here is that to learn that, and that's what took us the first year in my lab, to understand that our anatomy, actually, I had to go back to anatomy.
بس you were doing this في الانفلونزا قبل السارس صح؟ فكان عندك الادفانتج ده.
بالظبط. We started this with influenza. We had the whole team set up for flu, and once we had the pandemic, all the influenza activity was shut down, obviously, and we just used the whole setup now to address a very similar question. The vaccines are given in the same site, the same exact muscle. Most of our vaccine, the intramuscular vaccine, are injected in that muscle, the deltoid muscle in our upper arm. So to be able to do that, you have to learn that lymph nodes are really specialized, even in this part in our body. So there are some of them drain that part, the upper arm. Some are draining the breast area, and some are draining the back of the neck, for example. And so there are different parts, and everyone is basically, these are filters that just collect information. So you cannot go and sample that lymph node and say, oh, what's happening after vaccination? Because this lymph node has no idea, nothing happening there that's related to the vaccine. You have to sample those ones. We call them the lateral axillary one. Those are the ones that are connected to the muscle. So it took us a year to learn that. This is 2017-18 when I first started my lab. And then by the end of 2018, we were really good at it. Everything takes time to get good at it. And this is actually how it looks like. And this is from the ultrasound. I know many of my colleagues here, لو انت رحت الالتراساوند مع زوجتك او هي اي حد راح الالتراساوند عارف this is how it looks. And actually this is the needle going inside the node, sampling the part where we call the germinal center. This is the node. It looks like a kidney, but in that upper part and the outer part is where the germinal center happens. And if you just go with a small needle and come out, you sample that. And this is what radiologists do to look for metastasis. This is what radiologists do to look for lymphomas. This is what we did to look at responding B cells after vaccination. And then the astonishing part now, you can take these cells and do the very, very simple assay, which any immunologist really looks at. The very first thing is you want to know what are these cells. And to know what a cell is in our immune system, you look at the defining features are on the surface of the cell. And we use that with a flow cytometry-based assay. Again, I'm sure that many of you actually know exactly what flow is, but I'm just in case someone didn't really use that. It's a very simple procedure where you actually shine a laser on the surface of the cell, and then you have, depending on the reflection and of course, use certain antibodies that can bind to certain features on the surface of the cell. And then I can define the identity of this cell. And so what we do is when we take these cells from either blood or lymph node, we use that to really identify the different B cells. As B cells mature, they change these surface features. So we can actually track the changes by just tracking like the changes in 3 or 4 of those features. So once I know, oh, it expresses XYZ, then it must be a naive. Then it lost Y and Z, but now expressing A, B, and C. So now it's a germinal center without really needing to actually look at more than that. And that's something we learned through the years, how to do that. So the one thing that really was very clear after vaccination with the RNA vaccines, and I don't know if they are yet available in Egypt, but they are very immunogenic. So the lymph nodes, as you can see here, are huge. And the reason, obviously, there is no relative here, so there's no comparison. But I'll tell you in the next slide how it compares to other lymph nodes. So the way I compare it is using these flow cytometry plots, which is here. Here is regular influenza, and I'm going to walk you through this very slowly because it's just very important for the next few slides. You can take samples before vaccination and after vaccination. So this will be day 0, just before vaccination with regular influenza. And 2 weeks after vaccination. And even without knowing anything what BCL6 means or what CD38 means, you can tell what's different between the 2 images, right? The very difference is you have lots of more dots here, and each single dot is 1 cell. So basically, there are a lot of more cells for this particular phenotype. We call it phenotype, where it has different features in it. And that the cells that we know are called germinal center. Again, this is accumulated knowledge through the years that we know that germinal center express this and this. Now compare, this is after influenza vaccination. If you do the same thing after mRNA vaccination, this is 4 different individuals. You obviously can tell the difference. You can see how much more robust the germinal center is compared to influenza. It's almost 10 times more. And that's how RNA vaccines are incredibly immunogenic. This is the regular flu shot that I'm sure many of you take. It used to be Vaxera أنا ما كنت هناك، بس مش عارف دلوقتي ممكن يكون موجودة منتشرة أكتر. بس that's flu vaccination. It can make something, but it's nothing compared to what RNA vaccines are doing, and massive. It's 10 times. So we can actually see that because we do something very unique. We actually sample that lymph node over time. And that's, again, something that hasn't been done in humans before, as you can actually look at that reaction as it evolves over time. And what we saw, amazingly, is that the germinal center actually persisted for a long time. So again, we are looking at the germinal center here in this, and then you can see that even 6 months after second vaccination, you had this ongoing training happening, which is obviously remarkable. And to finish, we obviously studied these cells in detail, and we learned a lot.
سؤال specific شوية على الرسمة دي، في حاجة مختلفة في day 201 شوية عن كل اللي قبلها، صح؟ في حاجة بتقل، دي أنهي population؟
الدنيا بدأت تهدى. Basically, you're right. الجيرمال سنتر response بدأ يهدى. وذاتس normal. 6 months، 6 شهور بعد الفاكسينيشن. الدنيا بدأت تقل، وحاليا احنا we are studying the 9 months time point. So this is actually, this data is not published. this is
هو
going to go ahead.
في سؤال اتسأل متهيألي توقيته دلوقتي، وانا عارف ان انت هتجيله في الاخر، بس يعني نحضر نفسنا ليه، اللي هو طبعا انا اتناقشت معاك فيه برضه في اغسطس. ما دام الميموري كويسه كده، ليه في ناس بتقول ان في ري انفكشن بيحصل زي مثلا بالذات دراسات كتير من اسرائيل، وليه في ناس بتطالب بالجرعه الثالثه؟ والسؤال ده اتسأل فعلا على الشات يعني، فمش لازم تجاوب عليه دلوقتي، بس قول لنا
لا
عليه.
هقول لك، خليني اخلص السلايدز دول وهرجع للسؤال. سؤال مهم طبعا، it's a very very important and very relevant. ومش عايز اقول لك كميه الميديا كفرج ليه في الثلاث شهور 4 شهور اللي فاتوا. بس the last point that I want to make before I go for it is that now we can look at the bone marrow and remember that this is how the different isotypes in a bone marrow. This is influenza from the people. This is people, if you look in the same bone marrow, the same individual, but against tetanus or diphtheria, or use the spike protein, this is immunized people. So you have very nice induction of bone marrow plasma cells. So that germinal center response was very important to make a durable response. And the last point is this. This is how people are immunized with Pfizer. If you haven't, in red, if you haven't been infected before, you have a very nice peak, which is really coming from the short-lived cells, and then you have a very slow decay, and those are the antibodies coming from your bone marrow plasma cells. And finally, how that compare if you have been infected. If you have been infected, you are one of those. If you have been vaccinated only, you are one of the red dots. If you have been infected, then vaccinated, you are one of the black dots. And clinically, that population that have been infected, then vaccinated, are clinically doing much better than any other population. And that
طبعا ما
explains.
نتمناهاش لحد برضه يعني.
Now, it's not a public health control, but this is what happens when someone gets infected, mild infection. You have a really good memory, you have a good
لا قصدي لان حضرتك عارف ان في ناس كتير بالذات في بعض الاماكن في امريكا قالوا طب ما نعمل الكوفيد بارتيز ونروح نعدي بعض عشان نعمل المناعه وخلاص. وفي ناس طبعا ماتوا.
Yeah, then you are part of here. And are you really much better if you are part of this group or part of this, which is basically you have the infection, but for many different reasons, your immune system didn't click. And you have really low level, you are not protected. So that actually brings me to the really important point about the variants. كل حاجة اتكلمنا عليها لحد دلوقتي are assuming that الفاكسين اللي انت خدته والباثوجين اللي انت بنحاول ان احنا نعمل protection against are having the same features. انت basically بنعمل training to our immune system to recognize certain features, but you don't know. And we train them very well, as you can see. We train them very well in a germinal center to recognize certain features and recognize them very well. Now, what happens is, usually with influenza, is very clever, but we are seeing it more now with SARS, is that if the virus changes that spike protein, now everything you have been doing is basically cut in terms of efficiency. Again, if we make an assumption with numbers, if you made, for example, 100 antibodies against the spike, and those 100 antibodies, if we keep the same percentage, 20 of them are neutralizing, okay? And you have that, now that's your arsenal, that's what you made. Now, this is all measured against the original virus. The 100 and the 20, this is all against the original virus. Now, go to a virus that changed a little bit. That 100 will become 60, for example, 50. Depends on how much changes. And that 20%, that 20 antibodies, usually neutralizing antibody gets a major hit because they are really in a variable regions. So that 20 antibodies now became 5. So that's the problem with Delta. That's a problem with any variants, is that your major repertoire got a hit, and that's why you need. So someone will come and say, "Oh, do we still need to get? Are those 5 might be enough?" Yeah, they could be enough. They could be enough to prevent they could be enough to prevent infection or severe infection. They could be enough, mostly they are enough to prevent death or severe disease. But they're most likely not enough to prevent the very first entry step. And there will be some viruses that will enter and cause the infection and make someone test positive. And that's what we call breakthrough. It doesn't take much RNA of the virus to test positive. And that's a problem with variants. Why do we need a third booster? That question is, or a third immunization for people who got Pfizer or Moderna or the very strong RNA vaccines. I personally didn't get the third one yet. It's available now for anyone. You can walk in the streets and get your third shot. I don't think we need it clinically. I don't think we really need it. Especially it's made of the same virus, the original virus. Yeah, but I think it's not going to hurt, because what will happen is it just will elevate the baseline to a higher level, which can be good. I mean, if you want to prevent the not just the disease, but you want to decrease the instance where you have someone who has even virus for 2 days. If you have not been vaccinated, you will be shedding virus, for example, for 10 days, a week to 10 days. And this, again, all approximation. If you have been vaccinated but got a breakthrough infection because the antibody efficiency got low, you will be shedding the virus for 2-3 days. Again, you're still shedding virus. You probably could be infectious in that period. But if you really want to minimize that period and make sure that even if you get infected, you are almost not shedding any virus, so you're not transmitting the virus, then yes, get the third shot. And that always helps. Again, with epidemiological studies, you have to consider many other confounder factors and other. There are lots of players. Immune system is not the only factor there. As age,
human behavior بالذات عندنا
كل
denial بتاع ان خلاص
في كل حته.
الناس معتبره الفيروس انتهى.
I always say that it's amazing actually في كل حته، مش بس في مصر، في كل حته، في كل culture. ملهاش دعوه بالeducation، ملهاش دعوه بالreligion، ملهاش دعوه بالrace، ملهاش دعوه باي حاجه. It's amazing actually. You can go to the most well-educated places in the Scandinavian countries, and you get still the same trend of people that don't want to get the vaccine. Till you talk about natural infection, you can go deep in the US, deep in the South, and you get that. You go all the way in California, you'll get that. You get in New York City, you'll get that. It's amazing. There was in New York City, there was a demonstration. They closed the street because of the vaccine mandate. So that part is universal. I've never seen something so universal, like people having نظرية المؤامرة عن أي حاجة لها علاقة. It's a bit frustrating, but at the same time, you can't control it, so you really try to educate and hope for the best.
في سؤال تاني هنا بيطرح نفسه برضو من برضو معلوماتنا في الإيميونولوجي، إن برضو الأنتيبوديز ما هياش يعني طبعا الفيروس بيميوتيت وسريع، لكن برضو الجسم مش خالي من الحيل. فاحنا برضو عارفين إن الأنتيبوديز usually improve في السبيسيفيستي بتاعتها والأفينيتي خلال الانفكشن أو يعني البروسيس ده. فايه موقع الكلام ده؟ يعني the antibodies that we secrete later in the process من البي سيلز بتبقى usually they undergo somatic، يعني مش مهم الميكانيزم، بس بيبقوا افضل عاده. برضه هم بيتدربوا زي ما الفيروس بيميوتيت. وكان في بيبر قريب سمعت عنها استغربتها جدا، كانت اتنشرت في ساينس، بتقول ان في ناس claiming to be totally naive للفيروس و they still were able to، يعني البي سيلز بتاعتهم were able to adapt a little bit to coronaviruses. ايه تعليقك؟
It wasn't adapt. The paper gave it from a very good friend and from a lab in Harvard. They did really. It's a different. The question you just asked is just a very important one. أنا بقالي literally 3 شهور 4 شهور كل واحد في المعمل عندنا شغال على الموضوع ده بالذات. For me, that's the most fascinating part of our immune system. I talked about the Germinal Center, about the training, but I didn't get into details about how that training is improving our cells. I said they are better trained, but I didn't explain the details. I'm happy to go through this. That's the work that hopefully will be published next week. So it's a massive effort we did. We followed the lineages of those cells, and we followed them over time, and we actually can see them as they become better over time, and they can be affinity matured. So the same exact cell can.
So this could يعني ده ممكن يحسن شوية مشكلة الفاريانتس.
Yes, it is improving our, even if our immune system didn't even see any variants. Obviously, most of the people we are studying only got the vaccine, so they didn't see any infection. But our immune system is basically, it's almost by probability. If you are improving the binding and enhancing the neutralizing capacity of an antibody against the original strain, if that same target still conserved with the variants, of course you are, by following up on that, you are actually improving the enhancement, the response against the variants. It's just as a follow-up. So it's really not meant because we haven't really seen the variant, but it's a byproduct as you can think of it, or a bystander effect. So yeah, so that's the part for me is the fascinating part of our immune system, is that not only حتى لو أنت قاعد 6 شهور بعد vaccination you have done nothing, your immune system is actually improving slowly. And even if the number of the concentration of the antibody in the serum or in blood is actually going down, but actually what you are getting at the end is better quality. You can think about it this way, is that there are many things in our life where you actually, when you leave it to age, it gets better. Of course, you can think of it as getting wiser. جبنة. في حاجات كتير ممكن تسيبها وكل ما تقدم كل ما قيمتها تعلى. I think of our antibodies in the same way. They can be less in number, but are much wiser and much better and much well trained. And it's a lot of to be able to see that over time, and the small environment of the lymph node and how that selection happens is exactly what we are hopefully will be sharing next week. And that's the process of
إن شاء الله نشوف الفيديو.
how the transformation.
في سؤال من الدكتورة.
Can go over it. It will take us another hour or two to go over it.
لا شو في سؤال من الدكتورة ريهام صام عايز أسأله. أنا برضو شفت اسم دكتور أحمد شريف دلوقتي وبرحب بيه. أحمد شريف عطية ما أعرفش لسه موجود ولا لأ، من ضمن الناس من القسم برضو اللي مهتمين جدا بالانتيبوديز لديفرنت انفكشنز. السؤال من الدكتورة ريهام عصام كان في الأول بس أنا سبته دلوقتي عشان وقته حسيت إنه أنسب بعد البون مارو. طب إحنا هي سألت على البلازما ترانسفير. طب what about البون مارو ترانسفير بقى؟ يعني I know it's quite invasive بس would this يعني transfer memory as well?
That's a very interesting question actually, and I know we are trying to do some experiments related to that in mice, actually not in humans yet. It's fascinating about those cells is that biologically there's a lot of questions to answer about them, and it's just you can spend, and there are many labs already that this is what they do for living. Every 24/7 is thinking about how those cells survive. So fascinating part about those cells, once you take them out of the bone marrow, at least in mice, they don't really do well. They don't tend to be able to survive. They almost like, عارف لما يكون في حد عندنا في مصر، أنا والدتي نفس الشيء، إن لو خدتهم طلعتهم بره بيتهم they just everything goes wrong. لكن هم في ناس عم بيتحطوا في نفس المكان فترة طويلة, there's something about the place that makes them just love to be there. And if you take them out, even if you provide them with everything you think they need, give them all the riches, cytokines, growth factors, everything, they still don't do well. And for me, that's a fascinating question. It's a puzzle. How you can actually do that. If you take these cells out and put them in a new niche, new environment, you need to make sure that you understand what was actually making them happy in the first place, so you can provide it in the second environment. So right now, if you take plasma cells in mice and transfer them to a different mouse, they die very quickly. So somehow, if you, again, this injection them IV. So I don't know. We are working in that area. It's still very preliminary. We are trying to understand that. I don't know the answer. I wish if I know. This would be a fascinating question to address.
تمام. في كذا سؤال اتسالوا على البوسترز، أنا ممكن اجمعهم انا يعني. اول سؤال طبعا بيقول هو لازم يبقى فيه بوسترز ولا لا، وانا عارف انا وانت يعني شخصيا مش متحمسين قوي للدوز الثالثه، بس في اسئله منوعه بقى. هل مثلا البوستر يفضل يبقى لو دوز ثالثه تبقى من فاكسين من نوع مختلف، ومش يعني مش موديرنا الفايزر، لكن يا حبذا لو مثلا فيكتور بعد فايزر او فايس فيرسا. سؤال تاني، هل تبقى تتاخد كده امبيريكال ولا تتاخد بس للاولد ايج؟ وسؤال برضو كان من الدكتور خالد مصالحي، هل تتاخد بيزد اون انتي بودي تايتر او نيوتراليزنج انتي بودي ميجرمنت، ولا اي حد ياخدها سو اون يعني. مجموعه اسئله كلها كده. ده سؤال. وكان في سؤال تاني اتقال في النص، يعني انا جاوبت عليه بسرعه على الشات، بس عايز رايك فيه اكتر برضو. طبعا كان في برضو conspiracies في الاول بتقول انت هتحط الار ان اي في الخلايا فهتفضل خلايانا تنتج سبايك على طول. طبعا احنا we know that the spike is not enough to infect يعني، بس هل اي خليه ممكن تكسبرس الار ان اي سبايك دي، ولا only specific cells و very localized؟ يعني هل immune cells بس، ولا انت قلت ممكن المسل cells كمان؟ and how
Yeah.
far this will go؟
أسئلة كلها جميلة. خلينا نبدأ من الأول عشان دلوقتي أنا تقريبا نسيت السؤال الأولاني. هو السؤال الأولاني كان
البوسترز، السؤال الاولاني البوستر.
لو البوستر نفسهم كانوا من different company, different product or different vaccine platform. I think if anyone that didn't get the RNA, I will tell them get the RNA, Pfizer, because it's really robust. If you had the RNA before, we are seeing some differences with the vector when it comes to T cell responses, not really antibody. So I say why not? You just try the Johnson & Johnson if you have. You haven't tried, but if you had Johnson & Johnson, AstraZeneca, I tell people just go for RNA. The data are clear. RNA after vector is much better than vector. So RNA after vector, that for me is no brainer. So that's the first thing. It's really all of them. They are doing the same thing. They are delivering the spike. Whether you deliver the spike في شكل العلبة اللي deliver فيها إيه مش تفرق. You are delivering the same food, so which is the spike. So for me, it doesn't make a difference between the two. The point about the who should take it. No one should depend on antibody titer for that. You shouldn't really. It's such a measure that has no meaning because of what we all discussed. It really has no meaning. So don't depend on saying, "Oh, I have that, then I must be protected. I have no that, I must be not protected." I think it should be based on your risk factors, which are, your health status, your immune system status, your comorbidities, drugs you are taking, your lifestyle, your surroundings. It's very simple. لو أنا عايش مع والدتي يبقى لو أي واحد عايش مع حد كبير في السن, it makes no sense in it to actually even to accept any small risk, because even if you can take it, you can transmit it to someone who really can't handle it. So for me that's number. If you're surrounding your work, if you're working with really teaching, you are definitely exposed all the time, more than usual. I can some people can isolate. If you don't have the luxury to be able to isolate, then you are likely more exposed. So I would say don't depend on your antibody, depend on your situation and all of the other factors. Then the most beautiful question, which is really a fascinating one. So we have now grant from Gates Foundation actually to address that exact question. How much RNA, how long the RNA lives in our cells? And you are absolutely correct. The RNA will never give you the virus, obviously. The RNA will give you the protein, but that RNA even doesn't live forever. That RNA doesn't even get into the nucleus where all our genomic DNA is. So there is no integration events. There is nothing about. There is no reason to think that cell that was able to get the RNA or uptook the RNA, express the spike, is transformed in any way that the daughter cells that come out of this cell will have any changes. Obviously, that's being studied, but at this stage, there is no epigenomic changes in the DNA, in the chromatin in the cell, that will allow that to be inherited to the daughter cells. I could be wrong. It's being done by many groups, but at least we don't see a reason for that yet. The point about that the RNA is how long it, we actually don't know in humans which cells are the most efficient ones. We think it's muscle cells, but also antigen presenting cells could be the ones that doing it more efficiently. It's a hard question to answer technically, so we don't know the answer, and that's why we are trying it in mice first. But we are definitely, and not we and many other groups are much better than us than doing this, and they are doing. We are just part of a team that trying to evaluate that, and I know there is a lot of interest in that. So. Again, this is a new platform. There's a lot of things to learn about it, and I'm sure there will be a lot of data coming out about this one.
شكرا يا دكتور علي. احنا قربنا على ساعتين كده، وانا مش عايز اعطلك. طبعا انت في بدايه اليوم عندك واكيد في ميتينجز وحاجات في اللاب، بس هو في سؤال اخير اتسال على الشات، وبعدين هدي فرصه للدكتوره شيرين لو تحب تختم في الاخر. لكن في سؤال اخير اتسال، هو كومن وسريع بس برضو مهم متهيألي. يعني طبعا بينصحوا دايما ناخد الفاكسين بعد الانفكشن بشهر، في ناس بتقول 3 شهور، بس what happens if somebody took the vaccine during infection؟ وكان في دكتور محمد صلاح برضو كان raised his hand، فنديله الكلمه بعد السؤال ده. هو بس كان logged out and in again. بس هناخد بس السؤال ده الاول. اتفضل.
اللي هيحصل في خلال مش really it's a totally different environment. You know, draining lymph nodes بتاعت infection are these bronchial lymph nodes, and they are really have basically you are giving the antigen in 2 different areas. The only thing that you consider is the clinical, and again, I'm not a clinician, so I don't have any clinical training, as you know, pharmacy, صيدلي. I usually don't really, I usually refer to my clinical colleagues, which more, especially the ID colleagues, when infectious disease. Where the idea is to let if I'm talking from immunological principles, then I would say wait for 2-3 months after infection. Why? Because infection by itself is giving you a lot of memory cells, a lot of germinal center responses, a lot of products that you can actually want the vaccine to engage. So why don't wait for 2-3 months and get the benefit of that? But if you just get vaccinated and you know that you're productively infected, then you're just instead of having, it's like you're eating 2 lunches at the same time. So you're not really, you are definitely cannot take the best out of both meals. So you're just basically, you're overeating in a way. But if you actually just take one, enjoy it, let your immune system enjoy it, and then wait till you get rest, then you get hungry again. And that's the best time to eat again, in a way. That's a really weird way for putting it, but that's how I think it. أنا جعان شكلي.
شكرا دكتور علي. دكتور محمد صلاح طبعا ايمينولوجيست كبير، بيشتغل على برضه الفيرال انفكشنز والكانسر مع التي سيلز، فكان عنده تعليق او سؤال مش عارف.
بحاول اتعلم يعني. لا، هو تعليق في الاول بس. حاجه مهمه جدا ان الدكتور علي اللبيدي يعني للاسف الشديد مش عارف اسميها ايه، في هيصه حصلت مع الكوفيد، لان طبعا في فلوس لابحاث الكوفيد وفي نشر في مجلات متميزه. لكن دكتور علي اللبيدي مش حد غريب او فرض نفسه على المجال. هو بفضل الله سبحانه وتعالى ما شاء الله زي ما هو ورى، هو اوريدي كان بيشتغل على حاجه قريبه جدا، اوريدي ما شاء الله كان عنده نيتشر وساينس من قبل الكوفيد. فمش هو يعني حاول يقفز على الموجه زي ما بيقولوا، وهو ما شاء الله كان very well positioned زي ما بنقول هنا، او في وضع كويس جدا، وما شاء الله مستعد جدا ان هو يضيف اضافه علميه، وليس زي للاسف في جروبس كتيره قوي مثلا هو بيشتغل على كرونيك فيروسز وفجاه نط ويلا وايه يعني. للاسف الشديد يعني حصل دوشه علميه ممكن نسميها للاسف. بس طبعا الحمد لله مع الوقت كل الحاجات اللي مش مفيده قوي بتتفرز، وبيفضل الحاجات الثابته الحقيقيه.
للاسف طبعا، للاسف دايما الناس زي ما بيقولوا في الكلام كده، الناس اللي عندها كلام تقوله بتستنى وبتتروى، والناس اللي ما عندهاش حاجه بتتكلم بسرعه. فطبعا كل الدراسات اللي طلعت في الاول كان فيها يعني قفزات غريبه من ناس عمرها ما اشتغلت في الفيلد. بس طبعا
يعني زي ما حضرتك بتقول فعلا، فاما الزبدي فيذهب جفاء الحمد لله. يعني فعلا الحمد لله الواحد بيبقى سعيد وفخور جدا بدكتور علي وزملاء كتير قوي هنا ما شاء الله، يعني عاملين كتير منهم بفضل الله من قسم الميكروبيولوجي صيدلة القاهرة، يعني فعلا حاجة فخر كبير. عندي 4 نقاط سريعة جدا جدا عشان مش عايز أسحب الناس بعيد ولا أضيع وقت. دكتور علي طبعا تقريبا معظم الشغل اللي وراه اللي هو متميز جدا، وهو بايونيرلي أو واحد من رواد هذه الأنواع من الدراسات في العالم كله مش بس في أمريكا. بس كنت عايز أسأل، هسأله بالإنجليزي وبعدين أترجم بالعربي على طول. How well does the bone marrow plasma cells that you're seeing reflect in the blood؟ يعني في صعوبة طبعا إن إحنا زي ما أنت قلت إن في كل الدراسات أو في كل التايم بوينتس أو النقاط اللي بناخد فيها عينات إن إحنا نخش نجيب من البون مارو. فإزاي إذا ناخد عينات من الدم دي تقدر تعكس لنا زي ما انت شفت برضو كان في دراسات طلعت من الناس في المعمل عندنا لورا فيلا وناس كده، ان يعني بعض الزملاء حاولوا يشوفوا خلايا نوع معين من الخلايا ازاي في الدم يقدر يديني صورة او فكرة عن اللي بيحصل في الليمف نودز.
ده سؤال مهم. اللي احنا عارفينه دلوقتي ودي studies actually done very nicely. I'm not doing it in my lab but it's actually being done. ان احنا بناخد البلازما cells من البون مارو and then take the antibodies from the serum and sequence both compartments. الانتيبوديز طبعا proteins فالsequencing يبقى mass spec. والplasma cells you sequence the BCR, B cell receptor and the sequence itself. And yes, there is a great corresponding between the bone marrow plasma cells and the antibodies. So yes, you can actually use the antibodies as a surrogate for actually what's happening in the bone marrow. It doesn't tell you a lot about the history of those cells and the origin and all of that. So studying the cell is always gives you more. But yeah, that's why those epidemiological studies, you cannot really get bone marrow from thousands of people. You only can get that from blood, and that's because it's easier. But that's what we have been.
ده مين يا باشا؟ فلوريان كرامر ولا مين؟
There are lots of people who are really working on the serology side of things. It's we call this. There is and I need to make really important point about the first point you made. Yes, there is a lot of groups that wasn't even doing viruses that are doing a lot of COVID work. But بس عشان نديهم برضو ندي البنفيت للموضوع ده. احنا بنعيش في الفترة دي في unprecedented event. الايفنت ده ما بيحصلش يعني غالبا لكل الناس اللي على الكول دلوقتي او اللي سمعوني النهارده. There is really unlikely ان احنا هنعيش حاجه زي كده بعد كده.
يا رب.
It's historically it's unlikely. يعني انت فكرت فيها the last event that was with similar size كان من اكثر من 100 سنه. ف probability wise we are unlikely ان احنا نعيش حاجه زي كده. حتى اولادنا unlikely يعيشوا حاجه زي كده. ممكن احفادنا. فانا عايز it's a historic event. I mean it's unfortunate but it's a historic proportion of the event. مش كل الايفنتس بتحصل فيها ان كل scientific research in all different avenues all different areas get shut down and that's اما بشوف ابحاث كتير من الناس ما لهمش ما كانش ليهم في الموضوع. الحاجه الوحيده اللي بفكر فيها ما هو الناس دي كانت شغاله في حاجات كتير وكانت شغاله بس كل حاجه اتقفلت. why not use the resources that they already have to try to address question? Yeah, they are not the best in that question, but they are trying to contribute, and that's kind of my way of looking at it at this stage.
تمام. وستيل الناس اللي كانت داخله بالابروتش ده عملوا شغل كويس وده هيفضل، وستيل الناس اللي ما عملتش حاجات يعني قائمه. طب
ناخد بوينت كمان يا دكتور محمد بس عشان نلحق.
تمام. لا انا كنت عايز
4 و5 في مصر.
الفلو انت قارنت بين الفلو اللي هو يعني بروتين بيست فاكسين والسارس كوفي 2 فاكسين، فيعني زي ما بيسموها في امريكا ابلز اند اورنجز. هل شفت حاجه او عملت ستادي على بروتين والام ار ان اي فاكسينز في السارس كوفي او you aware of something like that?
Yeah, that's a good point. We haven't, and we are really hoping. We were hoping that Novavax. This is the only company that I know leading.
Yeah.
I mean, eventually we will do it. It will take time when you have to get the vaccine approved, and then it becomes available. So it will happen. it just will take some time from us to get access to that.
سريعا جدا عشان ما ينفعش معلش يا دكتور رامي امشي من غير ما عكسه في الموضوع ده.
لا طبعا عكسه
قلت I give you credit for that طبعا ان كل السكسسفul vaccines ما عدا التي بي فاكسين احنا عارفين انها بتشتغل عن طريق neutralizing antibodies تمسك في الفيروس زي ما انت قلت فتمنع انه يدخل في المكان بتاعه ويشتغل. فده فعلا. بس النهارده هل من وجهه نظرك في الكرونيك فيروسز بالذات اللي فيها تغيرات في السيكونسز كتيره زي الاتش اي في والاتش سي في اللي طبعا الماسنجر ار ان دلوقتي هيسرع الابحاث في المجالات دي قوي، هل تعتقد انه ممكن يشتغل بس بوان ارم بالبي سيلز من غير التي سيلز؟
No, أنا
بس خلاص
I was oversimplifying.
اكتفي اخر حاجه ان شاء الله
طب بس عشان بقيه الناس تتابع بس عايزين نشير لدور التي سيلز.
Yeah,
food for thought.
Yeah, T cells are very important. It's really there is no way that actually even the B cell response will happen without a helper T cell. It's they all go together, and even protection. I'm just saying that we know that what we can correlate the protection is neutralizing antibodies. It doesn't mean that when someone gets infected or vaccinated with anything, it doesn't T cells don't contribute.
Engage, yeah. They don't.
It's just we don't measure it.
Yeah. طب اخر حاجه which is a venue for collaboration بقى ونتكلم فيه مع بعض لوحدنا offline ان هو طبعا to study the equivalent T cells in the bone marrow as well, like for whether for cancer.
انتوا الاثنين تكولابوريتوا معانا احنا بقى.
بالظبط يا باشا ان شاء الله يعني.
It will be that will be يعني ده حاجه احنا اتكلمنا فيها مع الدكتور رامي ان I think there are lots of opportunities ان احنا فرصه ان احنا نتعلم.
يعني فكره الفاكسينيشن ريكورد ان انت عايز تعمل الريكورد بتاع كل ال previous memory B cells, but I think like doing similar for both, like not for both like the major CD4 and CD8 but also for the different subsets and like you know منها بقى زي يعني سوري احنا كده هنخش حته انثروبولوجي شويه زي علماء الاثار هنجيب تاريخ كل الناس ونبدا منه نقدر ان شاء الله نشوف يعني ده بس كده يعني بزرع الفكره عشان ان شاء الله نتناقش فيها وسوري اذا كنت خدت شويه وقت من الناس.
مبسوط
احنا نحب نسمعك طبعا. انا يعني كان نفسي نقعد اكتر من كده. انا متاكد ان كل الناس اللي موجوده ما عندهاش مانع نقعد اكتر شويه بس احنا واخدين قاعه مجلس الكليه دلوقتي واعتقد العمال عايزين يقفلوا بس طبعا you can always chat online. I don't think حتى لو احنا خرجنا ما اعتقدش ان يعني انتوا ممكن تتكلموا مع بعض لو تحبوا. كان في يعني معظم الاسئله غطيناها والسيشن دي ريكوردد وان شاء الله هنشوف طريقه نعمل لها ريليس. يمكن هنقسمها على كذا بس على كذا فيديو عشان الميموري يعني البرود باند. بشكر دكتور علي جدا يعني بشكل كبير جدا. انا عارف ان هو مشغول وهو كان وعدنا بتوك في المؤتمر وعوضنا عنه يعني كان توك بثلث ساعه عوضنا عنه اهو بساعتين. وانا عارف كويس جدا من خبرتي يعني في زيارتي ليه وزياره اي بي اي في امريكا ان الساعتين دول صعب جدا يتوفروا في اول اليوم كده. فانا بشكره جدا جدا وبالنيابه عن كل الناس اللي يعني اللي موجوده هنا بشكره. لو دكتوره شيرين دكتوره نورتان كمان بتشكره هنا على الشات. لو دكتوره نورتان او دكتوره شيرين يحبوا يقولوا اي حاجه في الاخر برضو. دكتور ايمن.
شكرا يا دكتور علي جزيلا. انا عايزه اقول لحضرتك حاجه. انا بس كنت متخوفه من محاضره الإيمونولوجي. انا بالنسبه لي ده من نوع من انواع الساينس اللي كان صعب بس انا محاضره حضرتك بجد فهمتني حاجات كتيره حتى لو ما كانش ليها علاقه بالكوفيد. انا فهمت البيزس كمان. يعني انا بس عايزه اقول لحضرتك انها كانت بالنسبه لي قيمه مضافه جدا وحاجه اللي حضرتك قلتها بصراحه برضو اديتنا كلنا امل. انا بس عايزه اقول حاجه بس اضافه ثانيه. انا الواتساب شغال عندي كل شويه انها ندوه رائعه ندوه يعني كل الواتساب. انا نفسي اوري حضرتك الواتساب اللي عندي قد ايه الناس مبهوره بالندوه طبعا. يعني انا عايزه اقول لك شكرا جزيلا واتمنى يا دكتور بعد اذن حضرتك ان يعني يبقى في ندوات ثانيه لانها بصراحه كلنا استفدنا بالعلم ده جامد. وميرسي جدا يا دكتور. ميرسي يا دكتور رامي يعني شكرا جزيلا. ميرسي يا دكتور رامي على إدارة الندوة جميلة جدا جدا جدا. شكرا جزيلا. ميرسي يا دكتور أيمن.
دكتور أيمن
قاعد ما سابش اللابتوب خالص طول ما احنا قاعدين.
شكرا يا دكتور علي وشكرا جزيلا يا دكتور علي وإن شاء الله يبقى في فرصة للكولابوريشن ما بين القسم عندنا ومع حضرتك يعني في
ده
اي
أكيد.
حاجه، يا ريت
ده
تفكروني
أكيد.
في اي حاجه زي كده. احنا يعني
أكيد. أولا
عملنا حاجه زي كده.
شكرا لكم جميعا، يعني أنا
شكراً
متشكر جدا،
جزيلاً.
حاجة جميلة. وأنا بإذن الله لو في فرصة إن شاء الله نفسي أزور طبعا، وبإذن الله لو في فرصة إن أنا هزور وأقول لكم إن أنا
لا لازم تشرفنا طبعا، اكيد
فده
10
ده
سنين
يكون
كتير قوي انك ما تبقاش جيت الكليه بصراحه يعني.
إن شاء الله نعوض الموضوع بإذن الله، بإذن واحد.
شكرا يا دكتور محمد صلاح برضو،
جداً.
يعني ان شاء الله نشوفك عندنا على طول، ويبقى في تريبل كولابوريشن بقى، ده تبقى حاجه عظيمه يعني. لو
شكرا
بي
لحضراتكم
سيلز
كلكم
وتي
بجد.
سيلز
يعني
مع
طبعا
تجارب
بشكر الله يبارك فيك، بشكر دكتور علي جدا، لان فعلا زي ما دكتور رامي قال يعني الوقت ده ثمين جدا، وهو CNN اخرهم معاه 10 دقائق، فطبعا هو كتر خيره ان
حاجه
هو
النهارده يوم تاريخي يعني
يدي
بالنسبه لنا.
الكليه ساعتين، كتر خيره. وشكرا
شكرا
لحضرتك
دكتور علي،
دكتور
الف
رامي،
شكر.
دكتور شيرين،
واذا كان
إذا كان محمد صلاح بتاع الكورة جاب 3 أجوان هاتريك،
فدكتور
علي
علي
جاب
جاب
أكتر
اكتر
من
من
3
3
نيتشر
نيتشر
بيبرز.
بيبرز.
ده شرف ليا والله يا جماعة، شكرا جدا ليكم، نشوفكم على خير دايما.
شكرا
شكرا دكتور علي، وشكرا للجميع.
مع السلامة.